Here we will describe the physiology of capillary transport. So recall that the capillaries are the exchange vessels. They are bringing blood in close proximity to all cells and all tissues of the body so that nutrients, oxygen, and wastes can be exchanged. So overall, we see that materials can take 4 different routes to cross capillary walls. The first is that they can fuse directly through the plasma membrane if the substance is lipid soluble. They could be pushed by either pressure or osmotic forces through the intracellular clefts or through the fenestrations of the endothelial cells. We also see that transport could occur across the plasma membrane by vesicles, which carries large substances like proteins. So in general, we see that fluid is pushed out of the capillaries at the arterial side of the capillary bed by capillary blood pressure, which is also called capillary hydrostatic pressure. This occurs because blood pressure is higher at the arterial end, where it's about 35 millimeters of mercury, than it is at the venual end, which is where it's about 18 millimeters of mercury. And in general, we see that fluid returns to the blood at the venual or the venous end of the capillary bed by osmotic pressure that's created by the high concentration of plasma proteins like albumin that are remaining in the blood. So let's take a look at how both of these mechanisms work in a little bit more detail. So we'll begin with movement of fluid out of the capillary bed at the arterial end. So we see that filtration occurs at the arterial end of the capillary bed because the blood pressure in the capillaries, represented as HPC in this diagram, is around 35 millimeters of mercury. This blood pressure forces fluid out of the capillary bed. The osmotic pressure, on the other hand, which is created by the protein, like albumin, remaining in the blood, creates an osmotic pull of 26 millimeters of mercury back into the capillary. This force opposes the hydrostatic filtration pressure that we just saw. We also see that protein in the tissues creates an osmotic pull of 1 millimeter of mercury out of the capillaries. We can add this pressure to the hydrostatic pressure of the capillary, because both of them are outward pressures, for a total pressure of 36 millimeters of mercury of pull out of the capillary. If we subtract the 26 millimeters of mercury, which is the osmotic pull of the fluid back into the capillary bed, that leaves us with a net filtration pressure, or NFP, of positive 10 millimeters of mercury. This positive indicates that the movement of the fluid is out of the capillary and into the tissue fluid. So on the other hand, reabsorption of fluid occurs at the venual end of the capillary bed because the net filtration pressure, or NFP, is negative, which means that fluid is being pulled back into the capillaries. So once again, we see that hydrostatic pressure of the capillary, abbreviated HPC in this diagram here, has dropped to 17 millimeters of mercury. This force, just like we saw previously, still pushes fluid out of the capillary bed. However, we see that the osmotic pressure of the capillary, or OPC, remains the same at 26 millimeters of mercury. This osmotic pressure pulls fluid back into the capillary bed, and we also see that the osmotic pull of the tissues, which is abbreviated OPIF in this diagram, is still 1 millimeter of mercury out of the capillary bed. So we can add the osmotic pull of the tissues to the hydrostatic pressure of the capillary, or the HPC, for a total of 18 millimeters of mercury of pressure, pushing fluid out of the capillary bed. If we were to then subtract the 26 millimeters of pressure of osmotic pull back into the bed, we end up with a net filtration pressure of negative 8 millimeters of mercury, which means that fluid will be pulled back into those capillaries at the venous or the venual end of the capillary bed.